Hello, my name is Kapil Bharti. I'm a tenure track investigator at the National Eye Institute at NIH, and I want to thank Thermo for this invitation to talk about some of our ongoing work to develop an autologous iPS cell-based therapy for macular degeneration. So just to get everyone oriented, we're going to be talking about the back of the eye. So this slide shows you the homeostatic unit in the back of the eye, the retina on top, the photoreceptors, and in between the photoreceptors and the blood supply is sandwiched a layer called the retinal pigment epithelium. For most of the diseases that affect the back of the eye, it's this homeostatic unit that is affected during the disease process. So if we take a close-up look at this homeostatic unit, see that RPE is really strategically located between the photoreceptor cells and the blood supply. And as you can see in this slide, that RPE cells are highly polarized. They have different structures towards apical side and basal side. On the basal side, RPE cells sit on a perinaceous Brooks membrane, and on the apical side, they have these apical processes that interact with the photoreceptor outer segments. I like to call them the business end of RPE cells. And the polarized structure of these RPE cells is maintained by these tight junctions between neighboring cells. By doing this, RPE cells mediate metabolite transport from the blood to photoreceptors and backwards. And it also helps maintain photoreceptor health and integrity throughout its life by all of these functions that are listed on this slide. So you can imagine something like this that is so critical for photoreceptor survival and health. If RPE cells are damaged, it leads to disease onset and sometimes in severe cases blindness. And the next slide shows an example of devastating disease, age-related macular degeneration, AMD, that is caused by damage of RPE cells in the back of the eye. Here you see on one panel how we all normal people with normal vision see, and on the other panel you see patients with an AMD vision. They are blind in the central part of their vision. That means anywhere they look in the center part of their eye, they won't be able to see. The disease affects more than 30 million people worldwide, and currently, at least for the dry form of this disease, there's no cure. So when we look at the back of the eye of AMD patients, this is what you see as how the disease works. On the top panel shows you normal, healthy retina, the retinal pigment epithelium, and the choroid. But in patients with the dry form of disease, see that the photoreceptors are damaged, are dead, and so is the RPE and the choroidal blood supply. On the central part is where the patient's vision is gone completely, where there are no cells left. But then going from the central part towards the RPE, there's this area called the transition zone, where photoreceptors are still surviving and RPE is gone. So that suggests that these photoreceptors are on the verge of dying, is suggesting that RPE dies first in AMD, and similarly leading also to choriocapillary loss. My lab works on various aspects of these disease initiation, how these RPE cells start to become dysfunction, how the disease progresses as the cells start to die, and also all the treatments, both small molecule driven and cell therapy driven treatments that we're trying to develop for this disease. Another advanced stage of this disease is wet AMD, in which the choriocapillaries start to hyperproliferate and they penetrate through the RPE, leaking fluid and blood into the RPE and into the retina, again leading to blindness. What we do at NEI is use 2D and 3D models derived from iPS cells to both recreate disease in a dish and to also treat patients, or to develop cell therapies to treat these patients who have this blinding disease. And I'll walk you through some of our ongoing work. So just again to recap, my lab will start with patient cells. Patient or healthy donors make iPS cells from the somatic tissue, make RPE cells, and use these RPE cells to develop either 2D or 3D disease models for drug screens. What we call a disease in a dish approach, where we study disease pathways, do pathway analysis, and identify and discover new drugs, and hope to bring those drugs back to patients. And then at the same time, we use RPE cells to develop an RPE-patch that we're bringing back to patients as a cell therapy. So first things first, how do we make these cells from iPS cells? So the top panel here on this slide shows you a pathway how RPE cells, mature RPE cells are made from iPS cells. And really for a process that we want to work very efficiently, because if we want to develop a cell therapy or disease models, we really optimize how these cells are made at a very high efficiency and reproducibly to really a functional and mature state. And this was really guided by developmental biology of RPE cells, making them go through various stages of RPE development, neuroectoderm, RPE progenitor, all the way to mature RPE using just combination of different growth factors at different time points. The bottom panel shows you that the protocol really works well. You see a dish full of pigmented, homogeneously pigmented RPE cells confluently present in the dish. The middle panel shows you a flow plot that's looking at one of the RPE markers, showing you more than 98% pure homogeneous population of RPE cells, a very high expression of RPE marker. And then it also shows you how reproducible is this process. We've done it over 34 different donors, both healthy donors, diseased donors, and in every single case, we can make RPE cells at a very high efficiency in this protocol. Now, the next panel shows you, the next slide shows you a monolayer of RPE, really polarized monolayer of RPE, in this case made on a PLGA scaffold. This comes later in the second half of my talk, that we're using this biodegradable scaffold to transplant RPE-patch in patients. But this slide really illustrates done here by scanning EM, how polarized, how homogeneously polarized this monolayer is. In blue, you see the scaffold that has been pseudo-colored. In brown are the cells. You're looking at it from a side angle. You see 3 cells with the nuclei here. But most interesting is this green structure. These are remember the business end of RPE cells, the apical processes. And I hope you can appreciate that throughout the patch, this really homogeneously polarized tissue is present. And then, of course, if we are, again, trying to develop disease models that would really replicate patients' disease and also trying to develop cell therapy, we need to make sure that the cells are functional and behave like native RPE cells. I'm going to show you just one example that iPS-derived RPE do display all the features of native RPE cells. In this case, we are measuring electrical response of RPE cells. And this just one assay shows you so many different genes and pathways are working correctly in these cells. Most of this data has been published. You can view that in a previous publication. But what it shows you is that trans-epithelial resistance, really a function of the tight junctions between neighboring cells that I mentioned in my introduction, are correctly formed. Cells present resistance to the flow of current. Trans-epithelial potential, of the cells, difference between apical and basal membrane resting potential, showing how polarized they are. About 4 millivolts here is again similar to native RPE cells. And then the ability of cells to respond to extracellular cues, which is reduced apical potassium concentration, something that happens when light hits the eye, or application of ADP that causes this depolarization pulse, leading to calcium release in cells and activation of chloride channels. All of those acids, all of those functions are intact in iPS-derived RPE cells, and they behave almost identical to native cells. So suggesting that these cells, what we have made in a dish, are really true RPE cells. Now, the other cell type that I want to introduce before we get on with our 3D disease modeling is endothelial cells, again made with a similar developmentally guided process from iPS cells. A much shorter protocol than RPE. In RPE, to make RPE cells would take about 10 weeks, whereas in this case, we can make endothelial cells in about 2 weeks. Bottom panel shows you staining for CD31 here present here on these cells, and again by flow, more than 97% pure endothelial cells. Also, a protocol very reproducible. We have done now over actually 10 different donors, both healthy and diseased, and we can always make endothelial cells at a very high efficiency. So then what we do is to combine RPE and endothelial cells into 3D disease model. We use a technique called 3D bioprinting. What it does is on top you see a 3D bioprinter which has two print heads, 2 needles that move in a very precise XYZ orientation that you can control. And we fill this up with something what we call bio ink, which contains the endothelial cells that I just showed you, pericytes, fibroblasts, And a hydrogel that you can see at the bottom panel now, it's printing, the needle head is printing a zigzag pattern in this case. And we can print any pattern we like, but this zigzag pattern allows us to make bigger vessels in the area that is printed, and the empty area that you see in between is that where angiogenesis happens, and so we can quantify how much angiogenesis has happened from this endothelial cells. So the way this process works is we start off with a biodegradable scaffold that I showed you previously, made of PLGA. Electrospun nanofibers of PLGA. The scanning EM shows you how the network of fibers allows cells to grow on top of it. So take the scaffold, flip it in one direction, and we bioprint the bioink on top, just like I just showed you. Flip it upside down, seed RPE cells on the other side, and let it mature for about 4 to 6 weeks, and the scaffold degrades. It's replaced by ECM that is made both by RPE cells and by the endothelial cells and fibroblasts on the other side, and it becomes akin to similar to a Brooks membrane that is naturally present between RPE and endothelial cells, or RPE and choriocapillaries in the eye. And about 4 to 5 weeks, the tissue starts to look really like this. This is one example of our 3D bioprinted tissue. You have RPE on top, and in between, the scaffold has been replaced by ECM, and on the bottom you see capillaries here labeled in green for endothelial cells and in red for pericytes. And you'll see a dense capillary network throughout the bottom panel, very similar to the choroid. Next slide shows you a movie in which a Z-scan, we're running through the choroid side of it, and you can see vessels of various size of lumen from about 20 to 50 micron running through the entire tissue, again labeled in green for endothelial cells and red for pericytes. So the tissue combined together looks similar to this. Is the RPE monolayer side on the left, labeled here for apical process marker acerine in green, that shows you how polarized the cells are, and E-cadherin in red, which labels the tight junctions between neighboring cells, showing you that cells are confluent and electrically tight. And then on the bottom panel, you see a vascular network here labeled, very dense vascular network labeled here with the fenestration marker PLVAP. So that is the fenestration marker staining was interesting. That got us thinking about whether these vessels actually are fenestrated. So for those of you who don't think about fenestration every day, this is in capillaries, it's a small hole in the plasma membrane of endothelial cells, about 70 to 100 micron sized pores that allow easy flow of macromolecules. And it's a hallmark feature of choroidal vessels, but also of many other vessels throughout the body. So we compared how our vessels look like to published literature. You can see on the left panel here, published fetal human choroid, which has its fenestrations in the membrane of endothelial cells. And if you compare that to what we see in our vessels printed from iPSC, you can see a similar pattern of fenestration throughout the endothelial membrane. So this now really opens up a new field, new topic for us, what really causes fenestration formation in the cells. And we know that these fenestrations are dependent on RPE monolayer, but what causes them, what molecules are required, what signaling pathways are required to make fenestrations, and what causes fenestration loss in diseases like AMD. So now moving on with really 3D disease modeling using this tissue, I want to show you the first example of the 3D tissue where we have just a wild-type RPE and wild-type endothelial cells. So here on top, you will see acerine again in green and E-cadherin in red around the cells. And as I play the video, you will see the vessels in the bottom again labeled in green and red, CD31 and PLVAP. So the video going in the ring from top to bottom. This is the vascular network coming back up, vascular network again, and then the RPE cells. So what we're trying to do here to really understand and learn more about the tissue, we're doing what we call a depth scan, in which we see what on the right side is the scale of the depth scan. If the vessels are green, they are at the choroidal interface. If they're yellow, they are the Brooks membrane RPE interface, and if they're red, then they're above the RPE. So in this case, if I get back to the point again, you'll see most of the vessels are green, very few yellow, and there's no red vessels. Now, if you contrast that to what I'm going to show you the next slide, where we made a tissue in which we overexpressed transcription factor STAT3 only in RPE cells, and we know that leads to increased expression of VEGF and IL-6 by RPE cells. And we want to see the consequences of that on the choroid. So really pay attention to the vascular network. Here you see again tissue going up and down, dense network. But when we do the depth scan, you will see how the tissue looks different from the wild-type tissue. Here, instead of a lot of green vessels, we have a lot of yellow vessels. That means they are all the way close to Brooks membrane. Now, if you pay attention to this white circle, actually just moved. So right next to the white circle, you see right next to this white circle, you will see there's one vessel that goes all the way across from the choroid to the Brooks membrane all the way into the RPE. So really, just like as I showed the top panel here, what happens in wet AMD, that one of the vessels has penetrated through the RPE on the other side. So it's really the model that we've been able to create of wet AMD in a dish. Now I want to switch to another model, which is the other kind of advanced AMD, the dry AMD. In this case, similar tissue, but we have stressed it with a complement stressor, which has been shown previously to induce drusen-like deposit formation in a dish. And you can already appreciate that the RPE looks fairly thick on top of this tissue. Now, when I play, you will see the vessels at the bottom in red again, and ApoE, the hallmark feature of drusen deposit, is labeled in magenta color. So it has this RPEs on top, and you can see ApoE deposits turning around. You can see the vascular network is really sparse here as compared to the wild type or the wet AMD tissue. Looks like the vessels are regressing and dying off. And again, as the tissue comes up, you can see this dense ApoE deposit, now again highlighted by these two circles. You can see one fairly large ApoE deposit and one kind of a condensation of many deposits. Actually, this one is almost of the order of 100, 120 micron, very of the size that is thought to be the stage when the disease turns into an advanced stage. So really, by both of these, just an example of making dry and wet AMD models using the 3D tissue. But then at the same time, we can use this technology to also discover diseases that can help undo or rescue some of these cellular endophenotypes. So this EM shows you that these ApoE deposits look real. They are similar to the basal deposits, basolateral deposits, or basolaminar deposits seen on the basal side of RPE cells. Currently, we are actually running a CRISPR-Cas9 genome-wide CRISPR-Cas9 screen to identify, to discover genes that are responsible for this drusen formation. So what we did is we ran a drug screen for proteostatic drugs that would actually rescue RPE cells based on the proteostatic stressor, and identified 3 drugs and asked if these 3 drugs that can rescue RPE cell could they also rescue formation of ApoE deposit. And one of the drug, this is I think the blue drug here, can actually significantly reduce the amount of ApoE deposits formed in this complement stressor. So in blue would be control tissue, and in orange is the complement stress tissue, which has a significantly higher number of ApoE deposits over the tissue. But when we treat the same tissue with the drug as we stress it, we see that number of ApoE positive deposits are significantly reduced, almost back to the control level. So we're very excited about this because this is, I think, the first example of a drug that can reduce the formation of ApoE deposits, so potentially could reduce the formation of drusen deposits. So perhaps it can be translated all the way to patients. So that's the first part of my talk, showing you utilization of 2D and 3D disease models to both understand disease and these pathways in a dish, but also to do drug testing and drug discovery, which can be potentially for potentially novel drugs. But then now in the second part of my talk, I want to switch gears and talk about cell-based therapy. How are we using this approach to develop a cell therapy for AMD patients? So going back to the slide that I showed you previously about this transition zone, where the photoreceptors are still surviving, but the RPE is gone, the idea people had for a long time was to transplant an RPE implant here in the transition zone and rescue the overlying photoreceptors from dying. And in fact, it has been surgically tried where people have cut out a piece of RPE from the periphery of the same eye, translocated it into the macula, and grafted it. And in a small number of patients, excuse me, where this graft has survived and implanted and grafted, they saw that patient's vision was stable for years to come, and the photoreceptors over the area of the implant were rescued. And that is indeed for us the guiding principle for our process to develop an autologous cell therapy. Rather than cutting it out from the periphery of the same eye, we want to make it from iPS cells of the same patient's blood, derived from the same patient's blood. So the next slide shows you how we converted our research-grade manufacturing process into a GMP-grade manufacturing process, starting with draw of blood on day 0, isolating CD34 positive cells, reprogramming them into iPS cells. We nailed down, or we noted down every single day how different processes were going to be done. The SOPs were developed to making the bank, to characterizing the bank. And then from there, setting up differentiation of 3 iPSC clones in parallel, going all the way to progenitor stage, and then seeding the cells onto the PLGA-patch, like I showed you previously. So this process takes about 6 months, excuse me. But we have been able to map out every single day of this process, what's going to be done, how the iPS cells are going to be made, characterized RPE cells are going to be made, when the QC is going to be done, and what the QC parameters are going to be to really have a go-no-go point as we're moving forward towards cell therapy or clinical translation of this product. At the same time, we have here on the right, you see some intermediate stages where we cryopreserve our tissue or cells so that in case the process fails, we can get back to one of these intermediate stages and start again. So the process, again, in a clinical grade setting works really reproducibly and robustly. This is an example of a scale-out differentiation on purification of this process. What we've done here is 107 different differentiation runs across 28 different iPS cell lines, performed by 12 different donors, performed by 4 different operators. In black, you see what was the purity of cells before we have enrichment steps at day 40, before the enrichment step, and we range somewhere from 90% to I think almost 40–50%. But after the enrichment step, we go all the way to almost 99% purity in pretty much every single case. And I should say that more than 80% of the iPS cell lines we tested were successful in this process, and 100% of the donors that we tested resulted in successful RPE differentiation. So we are fairly confident that as we move into the clinic, we will be able to make robust RPE cells from every single patient that we enrolled in our clinical trial. Now, another important feature as we are making cells for transplantation in patients is how do we qualify that the cells are good, that they're ready for transplantation? So to do that, what we did is we developed a neural network that would allow us to predict transplantable RPE products. So the way this works is that we use what is called quantitative bright field microscopy, and in this case we take images of cells as they were differentiating, and we develop a neural network that would analyze those images and compare that to the end result, which is again the resistance of cells, transepithelial resistance of the monolayer, and use that as a learning set. But then after the network has been trained, we can use the network to now start making predictions using a new sample. So the network will be able to predict which donor the cells came from and what was the TER, what will be the TER of the monolayer. This was one way of doing it, and the second way was while we were training the network, we also segmented borders of cells by just a simple membrane stain, and often which was hand-corrected afterwards to make sure that the network does recognize all the cell borders. So we retrained the network on hand-corrected cells. and then extracted all the cellular features, for example, cell area, hexagonality, number of neighbors, and then use those cellular features to again predict the same outcome. Can we have the cellular features correlate to transepithelial resistance or donor identity of the cells? So this seems to be working well. I'll show you one data point for this. Again, you have already seen what we did, a deep neural network in which we trained the network for RPE images and TER and asked it to predict the function of the cells after it was trained. Here's an example of one data set. Here we're measuring transepithelial resistance of 3 different types of cells. In blue would be cells that are not matured at all. We call them fetal-like. They're chemically kept unmature. In green would be cells that have been partially matured. And in red would be cells that have been fully matured, again, chemically. And our threshold for TER in this case is 400 ohms per square centimeter. On the x-axis is the measured TER that we experimentally measure on this monolayer. Excuse me. And on the y-axis is the TER that is predicted by our network. So first thing that you notice is that the network is almost linearly working similar to our experimental setup, that it can work the same way. Both the network and the measurement are linear to each other. That means they have similar results. And second is, again, the network is able to tell us differences between less mature cells to medium mature cells to fully mature cells. So we think this could be really a way of validating and characterizing our scaffold, our patch, RPE-patch, before transplanting it into patients, and really figuring out if the patch was transplantable or not. Because it's a relatively, relatively easy test to run in the GMP facility. So after we know we can make cells robustly and characterize them, we want to make sure that these cells are safe before we transplant them into patients. So we perform preclinical animal studies to test both safety and efficacy of our RPE-patch. So first we tested whether these cells would lead to any tumor or toxicity in the eye. And that's one of the really biggest challenge with any stem cell therapy, that the worry is that if there are any stem cell left in the final product, they could lead to tumor or teratoma formation. Here's an example of actually pure iPS cells injected in the subretinal space, leading to this massive teratoma, pushing the lens out, and you can see all the 3 germ layers identified in this teratoma. But in contrast, when we transplant an iPS-RPE patch, it just completely integrates into the back of the eye, doesn't lead to any tumor or teratoma formation. These red cells are stained for STEM121, which is a human marker. And as you notice here, you don't see the scaffold anymore because it's 10 weeks past transplantation. The scaffold has degraded, and the human RPE patch has completely integrated into the back of the rat eye in this case. To test the efficacy of this patch, we developed a model in pigs in which we wanted to see the efficacy of the entire 4x2 millimeter patch that we intend to transplant in patients. But as you may know that in pigs there is no genetic model for AMD. So we developed a laser-induced RPE ablation model in pigs. And we use a micropulse laser that uses a 532 nanometer wavelength to selectively ablate RPE cells, because that wavelength is absorbed by the pigment of RPE. They heat up and buzz. And as the RPE cells would die, photoreceptors would start to die because they're functional, their metabolic support is missing. And that's what you see in the bottom panel here, infrared image of a pig fundus. See a 7x4 millimeter, what we call a window defect, where RPE cells are missing because now the infrared light passes through, and you see that's why you see lighter color. And that's also confirmed by histology. You see Brooks membrane that is without RPE cells, and also clumped up RPE cells that are just sitting there. As a result, photoreceptors are starting to die. The outer segments are much shorter than they should be. So the goal would be to transplant an RPE patch right here and have it protect the photoreceptors above the area of the transplant. So keep in mind that we're transplanting human RPE patch in a pig eye and asking it to protect pig photoreceptors from dying. So we develop a tool to bring the patch to the back of the eye, tool has a tip curvature that fits the back of the eye. It takes the scaffold, the patch, only in one orientation. It's made such that surgeon can see the patch while he's delivering it to the back of the eye, and it's controlled by a foot paddle. So the next slide shows you a video of the surgery. As you can see, the pig eye has already been prepped. The surgeon is making the incision in the eye wall a little bigger to put the patch tool inside the eye. And in the next shot, you will see the tool. As the tip of the tool is a patch, RPE patch. You can see pigmented cells on top. The surgeon is aligning it with a cut under the retina, and the retina has already been lifted. The patch will be delivered under the retina, and the retina will be flattened on top of the patch, so it stays there, which we can confirm by a technique called OCT, optical coherence tomography, where we can see the back of the eye by reflectance coming from the back of the eye. Here by fast view, we see the patch here, and by side view, this yellow line shows you where the RPE patch is, and you can see the retina is fairly healthy above the area of the patch. Now, to really measure functional outcome or functional efficacy of the patch, we use a technique called multifocal EFG. Basically, we're measuring electrical responses from the back of the eye from the retina by shining multiple different light pulses. So wherever the light will hit, we'll get an electrical response from that part of the retina. For instance, if the light hits the healthy part of the retina, we get a response, nice and big and robust response. But if the light hits the laser injured part where we did not deliver the patch, we get minimal or almost no response, and that would be like somewhere here in the eye. But if the light hits the transplanted part, which would be right above this yellow line, we get a response that is in between the healthy part and the injured part, suggesting that the human RPE patch is able to protect the pig photoreceptors to some extent and protect the retina actually functionally from dying. And we can also corroborate these findings by histology. The next slide shows you same eye, two different areas, laser area with no transplant and laser area with transplant. You can see the transplant here marked with the yellow line. You can see this human nuclei labeled here, pigmented cells. Notice the thickness difference in thickness of the outer nuclear layer, the nuclear layer of photoreceptors. Above the area of the transplant, it's almost double from the as compared to the area where there was no transplant, suggesting that human cells are able to protect pig photoreceptors from dying and pig outer nuclear layer from getting smaller. We can also show functional integration of the patch. This is kind of a 3D rendering of the part of the eye, part of the retina. In red is, again, human cells labeled with STEM 121. And in green, we have labeled rhodopsin, spigrhodopsin. And you can see that rhodopsin has been, a signal is also can be seen inside human cells, suggesting that human cells have taken up phagocytosis to pig outer segments. That's a typical feature function of RPE cells, showing you that human cells are actually functionally integrated in the back of the pig eye. So all of this work is summarized and led to an IND. What I showed you is that we performed clinical grade manufacturing of the patch. I didn't get to show you that we actually validated this data from 3 clones per patient and performed functional validation of the patch. I showed you some of the pilot studies that led to testing the safety of the patch in rats and in pigs, and also the functional studies that led to GLP functional studies in pigs. So all of this work has been completed, and we're in the process of putting together the IND to be submitted hopefully in a couple of months to start the clinical trial right after that. So with that, I want to thank the entire team here, which you see is the cell therapy team that actually last year got an award from the NIH director for their efforts to develop a cell therapy product. And then here is the 3D bioprinting team, some of the members from the team, Russ, MJ, Haruchi, and Roba. Some of these people have been working on the 3D bioprinting project. So with that, thank you very much.